Advanced Dual Mesh Nebulizer for Cystic Fibrosis
by Jeev,
Arush, Nehal,
and Sajid.
So, what is cystic fibrosis? Cystic fibrosis is an inherited genetic respiratory disease in which thick and viscous mucus builds up in patient airways. It is due to a mutation in the CFTR gene, which leads to a broken CFTR ion channel, which traps solute inside the cell and doesn't allow for water to reach the outside of the cell and make mucus more viable. This can lead to choking hazards, which causes fatality. But over the past 50 years, mortality rates of the cystic fibrosis have decreased, yet 11,000 people still die from it every year in the United States. But many patients still struggle with symptoms, most namely being that of breathing problems due to the thick and viscous mucus.
Despite CFTR gene mutation having a major role in this CF patients, there is prospects of looking at different targets. Mucins, which are a group of proteins that are heavily responsible for mucus production, are one such target. Mucin production depends heavily on MUC5AC and MUC5B gene expression, both of which are represented as mRNAs, which are then transcribed into proteins. mRNAs are messenger RNAs, which are single-stranded as opposed to the double-stranded counterparts of DNA, and they're primarily responsible for transcribing DNA sequences into proteins in ribosomes. They contain the instructions for these specific proteins. We are targeting the MUC5AC gene and the mRNAs it produces because it minimizes the off-target risks that we may have or any side effects, while also maximizing our efficiency in dealing with mucin production. And the issue that we see here is that despite there being dehydration outside, the body produces mucus as a response. However, this fluid imbalance remains the same as a result of fluid building up inside rather than outside of the cell. So the body continually produces mucins, which continually promote mucus production, and in the end, this forms thick mucus that leads to mucus stasis and eventually suffocation. So our target, as we decided, is mRNA sequence that transcribes mucin proteins from the MUC5AC gene. This is because the cell's chromosomal DNA is no longer affected if we target mRNA sequences. Targeting mRNA minimizes that genetic risk that we might pose the cell, while also maximizing the efficiency and efficacy of that target. Besides this, mRNA is a way of completely bypassing the issue of crossing the nuclear membrane, which would pose an issue since genes and the DNA of the cell is housed within the nucleus, whereas mRNA sequences are found in the cytoplasm.
So here are our treatment goals. Our first and biggest goal is to reduce mucus buildup in CF patients and create relief. Our next goal is to deliver targeted and personalized treatment plans based on disease progression, doctor recommendations, and suggestions from our nebulizer. Finally, we want to monitor progress and adapt accordingly. CF patients deal with different levels of mucus buildup on a day-to-day basis based on how much activity they're doing, or how far their disease has progressed, or generally how their stress symptoms are feeling that day. So by monitoring this progress and adapting accordingly, we can create better plans for everyone and increase patient quality of life significantly.
So the way of targeting these mRNA sequences specifically is through a complex known as the RNA-induced silencing complex. This is also known as the RISC complex, and the RISC complex is used in our body to effectively deal with viral mRNA sequences. And these are foreign, and so the body will find and destroy these sequences by cleaving these mRNA sequences. What we can do instead is use our siRNA sequences, which are known as small interfering RNA, and those are typically between 21 and 25 base pairs and are double-stranded, to bind with this RISC complex and identify this mRNA sequence. This works because the siRNA is composed of two different strands, one being the passenger and one being the guide, allowing the siRNA to both find the RISC complex and also be read and destroy its complementary mRNA sequence. So here's a demonstration of that, where our dsRNA becomes siRNA inside of the cell. It binds with RISC so that it can identify its complementary mRNA sequence, which in this case would be our MUC5AC mRNA, and effectively destroy it so that it can't transcribe into mucins later.
So the first part of developing our therapy is to find a suitable siRNA. So first we used siRdirect to find potential mRNA targets and siRNA strands, and then we used siRNA to evaluate the efficacy. So first we chose 21 base pair strands that had optimal temperature resistance from siRdirect and plugged that into siPred. And from there we selected the most effective strand, which was actually this one right here with a score of 1.142, which the prediction model defines as very high efficacy. So looking at our legend, we find that the blue strands are the guide strands, which actually bind to RISC and help bind the target mRNA, and the red strands are the passenger strands and are usually discarded after binding.
So how can we ensure that these naked siRNAs, though modified, can actually enter the cell, because they themselves are not viable in doing so? We also have another issue, which is how will they traverse the mucus layer, which is extremely thick and has a host of difficulties to pass through. Well, the ways we decided to do that are the following.
So we actually wanted to load up our siRNAs into lipid polymer hybrid nanoparticles, or HNPs. And these nanoparticles have a polylactic coglycolic acid core and a lipid shell of DPPC, which allows for the survival of the siRNA in the human airway. And it can also withstand CF mucus in stagnant airways. And the lipid-based particles will also have very good cell uptake without receptor-based interactions due to their solubility. And the size of the particle allows for a lot of siRNA loading into the nanoparticle without creating overly dense or heavy nanoparticles.
And we saw the following study which uses similar delivery method and found that in cystic fibrosis lungs, which typically have polymicrobial infections as there's mucostasis where it can be drained, there was uptake in cells with HNMPs with PLGA cores, which shows that these siRNA delivery method could induce cell uptake, as seen by this. siRNAs, despite being modified, can survive outside of the cell, especially in CF lungs, for extended period of time. Also, they can't be uptaken by the cell, as there's no method for doing so and crossing such a thick mucus layer. So the way to bypass this issue, we decided to do the following.
So we chose lipid polymer hybrid nanoparticles, or HNPs. These are usually comprised of a polylactic coglycolic core and a lipid shell of DPPC. This will allow for survival in the human airways and survival in the stagnant CF mucus airways. And it also allows for uptake of the nanoparticles because of the lipid shell can enter cells due to lipid solubility properties. Along with the lipid shell is perfectly designed in such a way that we can have a maximum loading of siRNA without making the particle too dense or heavy to enter the area.
We can also see in a similar study conducted that these HNPs were used to have these siRNAs be actually uptaken by the cell. In this case, this is CF lung with a polymicrobial infection, which makes it especially difficult to traverse the mucus. However, they found that these delivery systems were in fact having sufficient cell uptake and therefore producing the response that we needed to see. As naked siRNAs typically have a short shelf life, we manipulated them to ensure that they aren't easily broken down by the cell's body's nucleuses and have a longer term viability and better binding to RISC inside the cell.
So some modifications we made were adding a phosphate group. So as you can see on the 5' end over here, phosphate groups in general, when added to naked siRNA, increase the affinity to RISC and allow it to bind directly and with a greater probability of binding to the RISC protein. We also added some M-methyl groups. So as you can see on the 5' over here and over there. And the methylation of nucleotides increases the stability and processing of the nucleotides by RISC, which is why we added them over there. We also kept the 3' group unmodified, as you can see over here, as modifying them leads to lower RISC affinity and therefore lesser chance that they bind to the RISC protein.
With the passenger strand, we added an amino group, as seen by this NH2 on the 5' side, as it leads to better loading into RISC, alongside the same phosphate and methylation that we did before. And we can also see here that in the 3' we added a C16 or a hexadecyl. In this case, modifying this 3' section allows for increased silencing potency in the passenger strand.
We rendered our own naked siRNA in order to see what it would look like, and in order to have a proper reference for what we were creating. So when we modeled our siRNA, we found that there was an overhang over here and over here. This overhang is actually pretty normal and is actually a defining mark of an siRNA. Unfortunately, we weren't able to get our modifications into our rendering of the siRNA. If we were able to modify them, which wasn't possible in most of the rendering sites which we used, including Chimera and Assembly2 siRNA modeling sites, the methyl group would look like bright spots on the nucleotides, and the phosphate groups would stick out of the two nucleotides at the end. And here's a closer up picture of the overhang for the siRNA. And here's a model. So here's our siRNA render for reference with the overhang. And here's an siRNA model which depicts what an siRNA should look like with the overhang at the end.
So now we're going to be moving on to our delivery model, which is an advanced dual mesh delivery system. And so this nebulizer, the key features are going to involve a dual mesh as opposed to the conventional model's single mesh, a temperature control system, battery powered, so then we can monitor patients outside of the hospital, and multiple sensor integration, so volumetric sensor on the display of the actual device. So as you can see here, this is our model. I'll just do a quick run through with our components. And so first we're gonna have the liquid mucus penetrating particle siRNA. That's gonna be the liquidified substance that's gonna be in our cartridge. Next, we're going to have the dual meshes, of course. Those are going to have micropores, which are going to allow the transferring of the liquid. Then we're going to have the power button, which you can see right there. That's the user interface portion. Then the PLA, polylactic acid, is what's going to be used to print out this device. Then we're going to have microprocessors within the actual device. The mouthpiece, as you can see there, a touchscreen, the touchscreen LCD display, the nebulizer that's within the atomization complex. And then this is the part that's going to allow for monitoring. And so the integrated spirometer will allow to see how the patient's breathing has been modified by our treatment. Then we're going to have the piezoelectric sensor, which is going to be integrated into the meshes. It's going to allow to detect volumetric output, the LCD display, and then AAD, adaptive aerosol delivery technology, which is going to allow us to basically modify our therapy based on the patient. So now let's take a deep dive into the atomization complex of our delivery system. First, we can see that the cartridge that stores the siRNA solution to nebulize will basically allow this solution to be prepared via temperature modifiers. Then it'll travel to the two piezoelectric meshes, and so these meshes will include micropores, and these micropores will allow only the tiny nanoparticles to travel through into the upward complex. And so we're going to integrate temperature sensors into this entire complex to make sure that our siRNA molecules are not being disintegrated, which is a common problem in all siRNA therapies. So as these particles travel through the upward complex, they'll finally exit via the mouthpiece, which is going to be directly modified for our patient. And this will allow for an easy delivery system. So now we're going to go over to the activation of the device and see a quick summary of everything that happens from the patient pressing the start button to the display summary. So first, the patient presses the on button, that starts the device. Then internal microprocessors will activate. That will basically allow the temperature sensors to get input from whatever's going on in the liquid cartridge. This liquid preparation then will cause the display to set to ready. That's when the patient knows that they can press the start button and allow for the nebulization process to occur. So then the two piezoelectric meshes activate. The siRNA molecule is released into the mouthpiece. The display sensors update, and then finally, the system basically terminates and prevents over-delivery. So, to summarize everything that just happened, we have a display summary with all of the sensor information, everything that happened in the entire session. Then we have, after the dosage occurs, the mesh self-cleaning cycle, and then we have the AAD calibration. Basically, adaptive aerosol delivery adjustments. These will allow the dosage to be optimized every time the patient, gets treatment. Because from our spirometer-based modifications, our calculations from the spirometer, we'll have to modify the dosage, we'll have to modify the output. So this is just a summary of our display. So this display is on the actual device. This is what the patient will be interacting with. This is the LCD display. This is a touchscreen display. So first we have our temperature, which is going to be measured in degrees Celsius. Then the dose delivery. There's going to be that liquid cartridge, the liquid that's in the cartridge. Basically, as it's being depleted, this percentage will decrease and show a continuous demonstration of whatever is going on within the actual device. Then we're going to have a time. So basically, based on the percentage, we're going to have a time. How much time is it going to take for this session to complete? Then we're going to have the nebulization rate. How much of the liquid is being converted to the aerosol form? And then the mesh status. So the two meshes, are they both functioning well? Then on the bottom right, we'll have any errors. So anything that's going on with the meshes, anything that's going on with the entire atomization complex. And then we're going to have the settings where we can modify the nebulization rate depending on the patient, and then the device battery.
So what are our next steps? How we're going to best serve our patients? So the main point of our therapy is to relieve symptoms and hopefully reduce the mortality rate. So we'd recommend coupling our therapy, which is not on its own 100% effective, with CFTR modulators, which are these small molecule drugs that target the CFTR proteins and help them function temporarily to reduce viscous mucus. So by combining both of these drugs, which alone do not have 100% efficiency, we can significantly improve patient quality of life.
So what are some limitations or shortcomings with this treatment method? One is that it's lung centralized. Since CF affects multiple systems, including the colon, the pancreas, and the lungs, we're only focusing on one component, which seems to be causing the highest rate of deaths, and that is the lungs. So the patient's discomfort and symptoms in other regions do remain untreated with this specific treatment. Another issue is that repeated doses are required, and also that this treatment method is not a cure for the condition itself, but rather a way of treating the symptom of mucus buildup and mucus stasis. So the patient is required to continually take doses for an undetermined period of time to deal with these symptoms. This condition can't be cured by any current treatments either way.
So in summary, our design was a mesh nebulizer loaded with compact siRNA meant to silence mRNA that codes for mucin production, specifically the MUC5AC and MUC5B genes. And we perform modifications to both the siRNA sequence and to the inhaler itself in order to optimize reception. We use specific AI algorithms in order to find appropriate mRNA sequences and in order to target appropriate mRNA sequences. So what are the potential implications for our design? So some implications for our design is that it allows for more manageable symptoms. It allows for less overexpression of the mucin 5AC gene, which allows for less mucosa buildup in the lungs. It also has the potential for greater quality of life. For all people with CF, it allows for potential for them to have less, to be able to breathe better, to be able to have less of these symptoms in their lives. And it also encourages future research into siRNA solutions as a possible treatment method for lung diseases, and furthers research into siRNA in particular. Here are references.
Thank you
for listening!